Yeah. Do I have to get rid of yeast or just call up the dermatophytes? It's almost like I knew what I was doing, huh? Yeah, that was on page 9 where, I think it was on 9. Have you found it? Page 9, just above superficial mycosis in that paragraph, the second sentence in that paragraph. Change that to fluconazole. Pardon? Yeah. Okay. Dermatophytes. What does that mean? Derm means what? Skin. And phyte? Plant. They're skin plants. Plants that grow on your skin. That's what they thought they were. Okay. I see I even put it there, and you didn't answer. And they're also called superficial mycoses. The dermatologists are a different kind of bunch. I don't know how any of you go into dermatology, and they use a lot in Latin phrases. Tinea is used as a synonym, means ringworm, but tinea really means the word moth. And how the two got connected, they think maybe in the way back several thousand years ago, or they thought that was a moth, looked like a moth growing on the skin. So anyway, tinea. So we have a variety of teneas. And this is tinea corporis, corporis meaning the body. There's big buttocks with a nice lesion. Tinea pedis, tinea of the feet. And these can either be very dry lesions like this, or they can be wet, weeping lesions like that. It's also called onychomycosis, tinea unguium, or the nails. There's actually a slight difference between the two, but not important at this point. It gets the nails. What you have to distinguish is a lot of old people get nails looking like that, and also certain diseases you'll get nails like that. In fact, I have a book that just talks about toenails. So all toenails aren't dermatophyte infections. This is an actual tissue section. They can make tissue sections out of nails, which I didn't know. And the dark purple staining are the hyphae of the, or mycelium of the fungus. These only have one, all they are filaments. That's all. Like Cryptococcus in Canada only occur in yeast, more or less. And these only occur as filaments, more or less. Okay? And the purple staining ones here in this section, it's another section. Tinea capitis is tinea of the scalp. What this is a woods light shining on the scalp. The one species, Microsporum audouini, used to be common in South Carolina when I first came here. And every school ran the kids through a dark room and shined this wood light on them. And when they see them with this, then they send them to the doctor because that species glows. For some reason, again, unknown, we rarely see that organism anymore. We still see Tinea capitis, but not with that specific species. This is not a good illustration, but this is jock itch. There, the testicles are pulled up. Here are two legs, and here's the lesion. You ladies don't have to worry about this for yourself. Anyway, Tinea barbae, the bearded area of the skin, has certain tinea infections. Tinea versicolor. This is common, supposedly in 25% of the population. I never tested that, but if you look around, your other three neighbors have it. I mean, or one of you have it out of your three neighbors. And it comes mostly in the summer. It's a brownish stain on the back, on the front, chest, or on the arms, the upper arm more frequently. Comes in the summer when you're sweating. And this is what it looks like under the microscope. These are the mycelial filaments, and these are the conidia. And this is referred to as spaghetti and meatballs in the lab, is what it looks like. And this is a wet mount. And mostly you see the conidia. They come loose very easily. Now, a lot of people don't care about classifying the dermatophytes this way, but if you think of them in this form, it helps you in finding where your patient's infected. Anthropophilic is anthro meaning man, goes from man to man. Now, it's rare, but the way it goes from man to man is very frequently it's an old grandmother in the house has the infection, she's taking care of the child, and grandma passes it on. The other is hats and combs. So it's not really a communicable disease, but it can be transmitted. So those are some of the things. If you're going to pediatrics, you're going to see, that's when you're going to see these diseases. And zoophilic is associated with animals. And if you're in family practice and the old farmer comes in, it could be him leaning on the fence, and he gets it from the cows or the horses, and they're species specific for cows and horses. The cow runs against the fence and leaves the dermatophyte there, and the old farmer leans on the fence, and he's got these lesions on his arm. And then the last group are the geophilic, in which the animals will roll around in the dirt and get it. And back on the zoophilic, cats and dogs, the pets. So you've got to ask the family if they have pets, and maybe they have to get their pet to the vet. Because if you don't treat the pet first, the people are going to just keep getting reinfected. These organisms appear worldwide, but there are a lot of the dermatophytes that a species will occur in certain areas of the world, which I'm not even going to go into now. But if you run into this later in your life and in your career, that certain diseases are some in Europe, some in the South Sea Islands, but they're specific for that area, that species. There are 3 genera. The trichophyton, microsporum, and epidermophyton. They are readily distinguished organisms. There are 19 species of trichophyton, and they infect the hair, skin, and nails. Each one of these genera have a different predilection. Trichophyton will affect all 3 of the dead keratinized tissue of the body: hair, skin, and nails. They are characterized by a white, fluffy mold colony. Molds look sort of like they're cottony in general. And they have the filaments that I showed you before. And they have these large, smooth, thin wall, pencil shape. And they're septate, usually about 8 septa. Thin wall, pencil shape. All the genera of trichophyton have that similar, but you've got to, they do vary within, but that's a generalization, and they vary in the color of the media, and then we use biochemical tests to identify the different species. These are some of the biochemical tests. They grow or they don't grow on the different medium. Trichophyton rubrum, it's, if the patient has an immune defect, it causes not just the hair or skin, but a large carrion or a large abscess, like, which I think I have. And in Charleston, at the medical school there, they run many, many dermatology clinics, and the mycologist there, who's now retired, used to keep track of all of them. And according to her findings, T. rubrum is most common, and blacks in South Carolina, or at least in Charleston area. And this is a carrion, K-E-R-I-O-N. They have big swelling in a patient's, not just the hair or the skin. It's a small mass. The microsporum species, only skin and hair, no nails. 13 species, mostly white, but a variety of colors. Now these are spindle-shaped, narrow at each end, wide in the middle, usually 6 to 8, and they're echinulate. You don't see it good in this photograph, but see these little spurs, little spines on the outside. Now they do vary from species to species, even in the microsporum, but in general, they all have that same shape compared to the others. And again, according to the clinics down in Charleston, that the microsporum canis occurs mostly in the whites in the Charleston area. Don't ask me why. Apodermophytum flacosum, there's only one species in this genus, the one organism, and affects the skin and the nails. Whoops. And they have these bifurcated hyphae. They're multiple. They only have about 4 cells, 2 to 4 cells, and they're smooth wall and that club shape, very, very typical shape. Griseofulvin. You remember what I told you about Griseofulvin? It's a very early drug. I think it was discovered in the late 50s. Came into use. Again, thought it could treat a lot of mycotic diseases, but it's only good now, not even for other tinea, but just for pretty much limited to nails. Used for skin a little bit. It gets incorporated into the growing tissue and prevents the hyphae from penetrating into that tissue. So you won't clear up the nail until all the infected tissue has grown out, which in fingernails can be 2, 3, 4 months, and in toenails can be 6, 12 months. It's a very difficult disease. Many people have it all their life once they get it. They've even pulled nails out, completely avulsed the nail, and then eventually it comes back again. What we're seeing now is a new drug, terbinafine, and that's Lamisil that you see advertised on TV, which so far, it hasn't been around that long, seems to be, I hate to use the word, but curing. If you remember, there used to be a lot of ads on TV calling Step Up to the Mic, and that's myconazole. That's an over-the-counter now. Tinactin's an over-the-counter. I think all these 3 are over-the-counters now. Griseofulvin, keto, itra, and terbinafine all need prescriptions to get that medication. Digger dermatophyte. You see these in all the magazines and on TV. That's Lamisil, or terbinafine, as its generic name. And then I want to leave you with this. It's an unusual reaction. It's called the id reaction. You can, the person will have this lesion on the feet, on the hands, and no matter what you do, nothing happens to that lesion. And you inspect the whole body. You can't isolate. You send the lesion scrapings to the laboratory, and they don't find anything. If you look at the feet of this patient who comes in, and usually it's the dominant hand. If they're right-handed, it's going to be on the right hand, left-handed on the left hand. And you examine their feet, and they'll look clean, but you do scrapings between the toes. Well, I'll show you how to do scrapings in a minute. And send that into the lab. They'll isolate the organism. You treat that organism, and the hand lesions will clear up. Called id reaction. Okay, that sums it up. We send to the lab anything, whatever's affected. If it's hairs, you pluck out the hairs or you can cut them off. And what's nice about these, you can wrap them in a piece of paper, put them in a Petri plate, in an envelope, whatever, and just send them in. And that's a good specimen, the lab. A good skin specimen is taken with a scalpel, scrape usually the edge of the lesion. You remember the fairy ring? That's the same thing. The edge of the lesion is the actively growing organisms. So you scrape the edge of the lesion and you put it in an envelope of paper, whatever, like this, and mail it in to the lab, or you could put it on a slide with Scotch tape. That's often done, and the lab will culture it and identify it. This is a neat little thing. When there was outbreak with kids, these are, I guess you wash your hair with those things and you rub them in your scalp. That's what they're sold for. But if you rub that in the scalp of a child that you suspect of tinea, and then they just put that onto the Petri plate like this, and wherever those tines touch, there's a colony of organism growing. So that's a good clinical tool. This is a hair shaft, and what I want you to see here is that the organisms grow in the hair shaft, and these are the conidia of the filamentous fungus growing on the edge of the hair. That's what it really looks like under the microscope. Okay, is that words of wisdom to sum up the lecture? Okay, now we're finished a few minutes early.